Okay, it's a great pleasure to have David on GEOTOP A giving his talk on I see interplay between DNA and mechanics and biological role even. So we are very curious to learn from you, David. To you.
Thank you, Enzo, and I want to thank you for your invitation to present here and to talk about this area, which is very dear to me. And in fact, I've been working in the area of DNA mechanics and topology since my graduate student years, on and off, and I find it fascinating and infinitely rewarding. The more we learn about this area, the more there seems to be to discover. So I'm hoping to convey some of that to you today. So I'm going to be focusing on the relation between the topological point of view and how we look at DNA, and then mechanical view, and how all of those things are reflected in the biological role of DNA. And my talk will be structured in three parts. The first will be, I'll be talking about primarily about DNA supercoiling, how that comes about from considering the topology of two closed curves, and the relation between supercoiling and elasticity, because the elasticity, the energetics of DNA, is an important component in supercoiling. Then in the second part, I want to take it from the equilibrium considerations into looking into dynamics of DNA, and including more than just the elastic energy. We're going to include also the electrostatics component in that part, and the motion of DNA in a fluid environment. And then in the third part, I'm going to be talking about the sequence dependence of the DNA elasticity and how we used that information to work out models of DNA together with proteins, and how we examine its role in transcription regulation. So just for those of you who are not familiar with DNA, the famous double helix is probably familiar to everyone, but we need to go a little bit more into the local structure of DNA. DNA is comprised of nucleotides. The pair within each nucleotide, you have nucleotide-base pair. You have two pairs of nucleotides bound together by hydrogen bonds. And they're always complementary, so C always binds to G, cytosine to guanine, and then A binds to T, that's adenine and thymine. But these bonds are hydrogen bonds. They're not very strong, and so the DNA can separate into two strands occasionally, but the two strands, the nucleotides in each of the strands of the double helix, they're held together by covalent bonds, and they're much stronger from chemical point of view. And both the covalent bonds in the backbones, in the two strands, and hydrogen and stacking interactions between the base pairs, which are fairly planar, as you'll see later on, they give DNA a rigidity, both with respect to bending and with respect to twisting, and that's an important observation that plays an important role in what the DNA does. So when you think of a closed molecule of DNA, the chemical nature of these backbones is such that the backbones can only bind to their own. In other words, once the backbones are closed up, they form two closed DNA ring, two closed rings. Now, many of you, or many people who have spoken at this symposium, have focused on the topology of a closed DNA, where you idealize DNA as a single closed curve in space, and in that aspect you focus on DNA knotting and linking. I'm not going to talk about that today, although I would like to just mention that the primary biological processes in which knotting comes into play is recombination, DNA closure, and replication, and that we have enzymes which can reduce the topological complexity of DNA. What I want to focus on today instead is the mechanics of DNA, where we focus on DNA as two interlinked curves in space. So we look at the backbone, the covalently bounded backbone, and idealize it as a curve. And in that sense, you know, there's a familiar topological constraint that you can associate with such structure, which is the Gauss, the linking number of the two strands, and the linking number, of course, can be computed in various ways using either the Gauss formula, which is a double integral, over the two curves involving the tangents and the locations of the curves. But we can also look at the linking number as 1/2 of the sum of the signed crossings in a planar projection of one curve crossing over the other, where the signs are by convention assigned either positive crossings or negative value, +1 or -1. And that way we can reconstruct the linking number of the closed molecule. As you can see on the right side here, the linking number would be equal to 8. Now the linking number by definition is takes only integer values, and it's a topological invariant. So for any closed molecule, as long as we don't perturb or cut the bonds within any of the strands, this linking number would be preserved under any deformation. Now, there is an important formula, though, which relates this number to two quantities that we can use to describe the structure, which are not topological invariants, and one of them is the writhe. The writhe is simply can be defined as the same Gauss integral, but integrated twice over the same curve, or we can define it using, again, the computation of the crossings of the curve with the same convention of the crossings of the curve over itself, but now we have to average these crossings over all planar projections of the curve, not just single projection. And now the writhe is you can think of it as a measure of the chirality of the structure. So if you have an achiral curve, if that curve is a reflection of itself, can be converted, then the writhe will be 0. But if that curve contains helical parts, like here, there's a helical part on the curve A here, or a helical part on the curve C, that helicity or chirality induces usually induces a non-zero value of the writhe, and the left-handed and right-handed structures correspond to either positive or a negative value of the writhe. So you can think of a writhe as a measure of helicity of the curve in some sense. And then there is a second quantity. When we have two curves, we can measure how one is rotating about the other, and this is called the twist. The twist, you can describe a local rate of twisting of one curve about the other by using a director to span from one curve to the other, this director D, and then measuring how D changes alongside of the curve, and then integrating that quantity, divide by 2 pi, and we get a twist. Now these two are connected together by a theorem called, which was actually first discovered by biologists, but then it was later proved for spatial curves, and then in more generality by Calogaran and White, and this is the famous Calogaran and White Fuller theorem, I think it's now called, that the linking number of one curve of two curves is the writhe of one curve plus the twist of the second curve about the first curve. And now, how does that relate to DNA? The DNA, when thought of as a closed molecule, there are two ways you can think of it as being reflected in the DNA structure. You can either untwist the DNA. Let's say the DNA has a certain amount of linking number, you untwist it locally, and what that means, because the linking number is preserved, it will be reflected in a deformation of the axial curve that gives it a writhe of the same magnitude but opposite sign. Or you can change the linking number by either by using enzymes or by nicking and by cutting one of the strands and rotating the other about the axis, and that can change. That will result in change of both writhe and the twist. but those changes will now be in the same direction. And why this happens is because of the energetics of the molecule. Now, there are some biological events where the supercoiling happens in regularity, and for example, that's a transcription in which one enzyme travels, the RNA polymerase travels on the DNA, and it has to untwist the DNA, and as it travels, it creates positive supercoiling. So this is the structures with positive right in front of it and negative supercoiling behind it. You can see it in DNA replication, where the DNA is copied into two strands, where the motion of the replication bubble also induces change in right, or intercalation, where molecules insert themselves in between DNA base pairs, and that way they untwist the DNA, and which generates the ride. And this supercoiling is regulated by enzymes which are called topoisomerases. And here, an example, type 1 topoisomerase does what it does. It cuts one of the DNA strands and then lets the DNA swivel around the other uncut strand, and that way relieve the linking number by rotation. So to understand that mechanically, what people have been doing is use elastic rod models for DNA. So the simplest elastic rod model comes from Kirchhoff's theory of elastic rods, and the assumptions are we have a uniform homogeneous elasticity, naturally straight configuration. So we can describe the mechanics with two moduli, the bending modulus, which is constant throughout, and then the bending energy depends on the curvature of the axis of this rod, and then the twist density, where the energy depends on the twisting modulus, constant again, and the twist density along the rod. So the total elastic energy is then the sum of the bending and twisting energy. And one can use the calculus of variations with appropriate constraints like the rod closure and the constraints of imposed linking number to derive equilibrium equations for the axial curve of the rod and the twist density that you can see on the right. And in equilibrium, the twist density is actually constant, and it happens to be a Lagrange multiplier in this biaxial formulation, and the force also comes out as a Lagrange multiplier. Now, the beauty of this approach is that you can actually find explicit solutions for the tangent and even integrate that to find explicit solutions for the location of the axial curve in terms of elliptic functions and integrals, which I'm not going to show here. But those solutions depend on 7 parameters, which are easily associated with the curve itself, and there are some integration constants. So with that in mind, we can now look at the shape, the space of different shapes that an equilibrium configuration of a rod with an imposed constraint of a linking number can take. And we usually draw them on a bifurcation diagram like this one, where each line or curve here represents a set of equilibrium configurations. The right is on the horizontal axis, linking number on the vertical axis. So by increasing the linking number, we're moving along the curve in the vertical direction. Now, this first branch of configurations would be the trivial branch, where all the configurations are circular, where it's simply increasing the twist density, and they're stable up to the first point of bifurcation, which is A here. And then you have a branch of bifurcated configurations of the figure 8 shape branching off of that. And then you have higher order bifurcation corresponding to 2 or to 3 or 4 or 5 different maxima, local maximum of curvature within. Is there any question? Okay, now what happens if once you follow these branches up to the point of self-contact, ignoring the self-contact, you would pass into the situation where you have knotted configurations of the torus knot type. The main observation that people have made here is that all of these configurations, they do not involve self-contact, but unfortunately they are all unstable, and this has been observed already in the 80s. So if we want a structure of a DNA with some supercoiling that would not be that would not be unstable, it would be stable, that structure will require self-contact. So we need to be able to include self-contact in our model. So we're going to include the possibility of external forces acting on the rod at the places of contact, which will come. These external forces will come from another segment of the rod. So one segment, let's say at a location S star, will be in contact with a cross-section or a segment at a location S, and there is going to be a contact force F between the two. So if we include this, we need to include some geometric conditions for the contact, and we need to include the contact condition as an external force in our balance equations, and those would be in the balance of forces, those would be included here. So the force, for example, is no longer constant throughout the structure. It's only constant within a segment between points of contact, and then it takes a jump once you go through the contact, and then it's constant again between points of contact. And these jump conditions have to balance out for the two segments that we look at. So now once we include the contact, we get a new continuation of that first branch that I mentioned from the figure A. They're unstable until you reach a point of contact in the middle, and then at the point of contact they start deforming, and they deform until you reach points of two points of contact, then three, and then at the point A4 you actually have a line of contact here. Now, as opposed to DNA, the one thing I should point out here is we're using linking number as a continuous variable, not as a discrete integer. It's still a topological constraint, but you can think of it as, imagine you cut the rod open and then you rotate it, but you don't rotate it all the way around by 360 degrees, but only by a certain angle, and then you divide that angle by 2 pi. That would be the non-integer amount of linking number you've added into the rod or removed from the closed rod. It's the angle of rotation of two cross-sections before sealing divided by 2 pi. So now the various conditions have been developed to evaluate the stability of these configurations, but the main thing is, as long as the linking number increases with right or vice versa, the configurations are stable here. Now we can look at the second branch, and we can observe also the points of contact here developing at once we branch off of the main branch. We branched off and through unstable configuration until the point of contact. Now there are 3 points of contact actually appearing here, and at each of the points of contact we are now going through configurations with a D3 symmetry up to a point P, which is a point where those configurations lose stability. The branch continues, but those configurations are no longer stable here. There are stable configurations on other branches with contact, which break the symmetry. So you break it from D3 to C2 symmetry, which is just 180-degree rotation about a single axis. And then you also have examples of configurations here which have no symmetries whatsoever. So the bifurcation structure becomes quite rich, even for just this branch. And then you can imagine all of the other branches, they come off with their own types of configurations and other types of stabilities. The one thing we can take away from this, though, is that we have a possibility of multiple equilibria coexisting for the same value of linking number. So a supercoiling of DNA, supercoiled structure can exist in multiple equilibrium states for the same linking number. And here are actually examples of observed structures for the structure A, which will be the continuation in the first branch with 2 loops, and the structure B. They're both local minima. The B would be with 3 loops, both local minima, and in between you have saddle points which describe configurations with which are barrier for transitions between the local minima. And because we can evaluate the energies of these structures, we can also compute the height of this barrier, and these would be the numbers for a particular size of the segment, and in KT, which is a unit of energy that's commonly used for DNA. So, intuitively, you can think of it as one local minimum going over a saddle node, and dropping to another local minimum. Now, so far I've been talking about the static analysis of configurations. We can also do the same for knotted configurations, and we've done a study of a trefoil knot. The diagram, as you can see, is much more complex. We were not able to continue and connect all the branches together because of numerical difficulties. There are also other things happening. For example, this configuration, which seems to be the minimum energy configuration, has contact all the way around this circular. The circle here, we have various points of bifurcation. The takeaway point here is that all the configurations which are stable, they require self-contact. So with this type of theory, we can address the equilibrium configurations. We can do analysis, for example, derive stability conditions. But getting to analyze the full space of equilibrium configurations is prohibitively expensive, because you have to analyze and locate every point of contact, and that with numbers of contact, that grows the number of equations in your system. Even though you're solving just algebraic equations, that becomes prohibitive for large and complex configurations. So this pretty much concludes the first part of my talk. So if there are any questions, I can address them now.
I actually have a little question, David. As we grow with complexity, evidently we have to find out a rough measure that relates, say, information about energy with configurational, space configurations.
Right.
Evidently, I saw from your first analysis that trefoil crossing number doesn't help, because you have between stable and metastable, no change in crossing numbers. Evidently, energy is related to bending. I guess it's bending what is talking here.
Yes.
So
yeah, it's bending
could you
and
say,
twisting.
say it again, bending?
Bending and twisting energy. As opposed to, there are some researchers who work primarily in knot energies, and they focus on bending energy. Now, when you include twisting energy, this is what gives rise to all these supercoiled configurations and large numbers of contacts.
So my question would be, if you go to, say, the two locally stable configurations, so to speak, how much twisting energy would trigger that? Or if that is, because my intuition is that it would amount to few percentage, not so much. I mean, is important, but compared to bending is another order of magnitude. Would that instead trigger these local states or not?
That all depends on what your, let's see, you can measure the amount of twisting energy on this graph, or the amount of twist by departure from the diagonal. So you're right. If you look at these configurations, the amount of twist within those structures is fairly small compared to the amount of ride. It's order of magnitude smaller, I would say, especially up here, for those plectonic cases. So perhaps it would not play much of a role, but on the other hand, you have to think about if you eliminate the twisting energy altogether, how would you impose the linking number constraint? You can think of it as another problem where you minimize the bending energy as a function of ride. In fact, all of these equilibria are also equilibria of just the bending energy at a fixed value of ride. So that's another way to think about it that avoids thinking about linking number and twisting energy altogether.
Thank you.
David, can I ask a quick question related to this slide that you're showing?
This one?
So, yes, this one. So, following up on Renzo's question, it's related. Is this diagram applicable to any combination of, let's say, bending and twisting rigidity, or is it for a specific choice of the two?
This is for a specific choice of the ratio of A to C, so the bending to twisting constant. And what happens, you can actually analyze this very nicely. If you change that ratio, the only thing that happens is these branches will move up and down along the linking number. So they're going to be, they deform in such a way that the location in the ride location stays the same of the point, but it moves up or down. So you can think of it as stretching things in the vertical direction away from the 45-degree line. And we've discussed that in one of our papers. So the ratio of bending to twisting is what determines the positions of these curves. Okay, so I think I'm going to go on then to the dynamics. So if we want to analyze the dynamics of DNA configurations and DNA supercoiling, then we need to include some other physical phenomenon. So we still include the bending and torsional rigidity, but we also include electrostatic repulsion, because the DNA is negatively charged molecule and it will repel itself locally. This is similar to the Mobius potential for those of you who work in knot energies. And then we embed it in an incompressible fluid and analyze the low Reynolds number flow of that of the structure within. So we use the same description of the mechanics, except perhaps here in this work we've generalized the mechanics, so we include two bending moduli and twisting modulus, and then we include possible shearing and stretching within the structure. Again, we include self-contact because the electrostatic itself will not prevent hard contact of the DNA. And for the electrostatics, we idealize the structure. We move the charges away from the backbone where they're located in phosphate groups. We move them towards the axis and place them on the axis of DNA. And instead of just a standard Coulomb interaction, we use a screened interaction, which is the linearization of the nonlinear Poisson-Boltzmann equation that has been reduced to a Debye-Huckel screening term. So here, the electrostatic force depends on reciprocal of the distance of the two charges, but also is multiplied by the exponential of negative Debye screening parameter times that distance. So it decays much faster than it would in the Mobius energy type. And this is because in vivo, in the fluid, there are ions, positive ions floating around that will condense around the DNA, and they will screen the electrostatic interaction of that molecule. So for the fluid, we use incompressible Navier-Stokes equation, and we solve the whole system numerically using generalized immersed boundary method. So this is a method where you represent the DNA structure with a grid that's moving in space, and then you have a fixed grid represented by these by the black lines for the fluid. So what you do is you want to solve the fluid on the fixed grid, but then the DNA is moving around and you need to incorporate the forces that come from the DNA and act onto the fluid, or vice versa. In fact, the DNA is carried along with the fluid. We assume no slip boundary conditions. So it's mostly the forces that the DNA generates onto the fluid that need to be moved, projected onto the fluid grid points, and we do that using these discrete delta functions. So any force, the force coming from the elasticity, the force coming from the Coulomb interactions, the moment is converted into a force, and the electrostatic and the contact, they're all converted into forces that are projected onto the grid points through this delta function. And then we saw for the equations of the fluid, the Navier-Stokes with the force projected from the DNA. We use finite difference scheme with upwinding for the fluid. And the beauty here is that one can decouple the system into 4x4 systems when you transform into Fourier space into n cubed 4x4 systems where n is the discretization of the cubic domain. We use cubic domains. So then the algorithm works in such a way that first we compute the force and the moment exerted by the rod onto the fluid using the balance equations for the rod. Then we spread it onto the fluid using the discrete delta function. We update the fluid velocity by solving these Navier-Stokes equations discretely. We interpolate the velocity at the discretization points, and then we propagate the fluid and the rod along with the fluid. And we check the CFL conditions to see if the time step is sufficiently short to prevent any blowups. And then we repeat that. Now here's an example of one simulation. For these examples that I'm going to show, we started with a circle of 600 base pairs, DNA minicircle, and we put a certain amount of supercoiling there, excess length equal to 6. So we rotated 6 times before sealing the circle. And we use either 0.1 molar of salt, which is considered to be high salt, neutralizes a lot of the charges, or 0.01, which is a low concentration of salt. Now, as you can see in the dynamics, I'll try to start. What happens is the DNA deforms quickly up to a point of first contact, and then you have all other points of contact developing along the rod with the DNA, and then you have slow sliding into the final equilibrium configuration. And the decay is exponentially slow at the end, convergent. The red dots represent markers, fluid markers that were originally located alongside of the DNA. And what we were interested in here was looking at how electrostatics influence the limiting configurations, and also what is the balance between, we've seen in the previous, at the end of the previous section, that I showed that there are 2 different equilibria, the one with 2 loops and one with 3. So we would like to know here dynamically what is the likelihood that the configuration will evolve into one or the other. Now this is a deterministic behavior, so it's a repeatable behavior. But on the other hand, in order to get the DNA to deform initially, we know that it's an equilibrium with uniform twist density. We need to perturb the twist in order to make the DNA move away from the initial circle. And depending on how you perturb the twist in the initial configuration, you can actually control the path that the DNA will take and the limiting configuration towards which it goes. So on this picture, we did not change the perturbation. We only changed the linking number, initial linking number of the ring, and the ionic strength, the strength of the neutralization. So the first row is when you have a lot of neutralization, and that allows for a high degree of contact within those shapes. And you can see that for large numbers, like linking numbers 4, 5, and 6, you have a lot of plectonymic we call this plectonymic type of a structure where you have the helicity with a loop at the end, developing within those structures. Now, for small screening, that means a lot of electrostatic repulsion. The DNA does not come nearly as close to itself, and you have configurations which do not show this plectonymic supercoiling. So there's a clear influence of the electrostatics on what type of a configuration you have at the end. Now, here on this slide, we analyzed, starting from the same configuration, if we do perturb the initial twist variation. So we don't change the axis, we only change the variation of the twist density, keeping the linking number, initial linking number the same, namely 6. And by perturbing that initial twist variation, we can achieve multiple locally stable equilibria as limiting states for the evolution. States with the 2 loops, 3 loops with various types of symmetries, 4 loops, or even 5 here. And they have different energies, different final energies. And just like Enzo pointed out, the twisting energy is much smaller than the bending energy for these configurations. The electrostatic, though, has a lot has a large magnitude. Although really the important thing is here is the relative difference between electrostatics between the configurations. Now the configurations differ in right. So our conjecture here is that by varying the perturbation, we might be able to achieve, or should be able to achieve, any locally stable equilibrium configuration within the system, but we have not been able to find any proof of that statement yet. Now, in terms of the dynamical evolution of the structures, you can observe the trajectory of the linking of the energy and the writhe, and this would be the total energy here, the writhe, and the kinetic energy. And what we see is that the energy, of course, decay is monotone because the primary driving force here is friction and viscous drag of the DNA within the fluid. The writhe, though, the evolution of the writhe need not be monotone, although it's fairly uniform. The important thing is that the kinetic energy is definitely non-monotone. So we have fast evolution interspersed with slow evolution. So we have transient states here in the dynamics, which these local minima of kinetic energy, which correspond to transient states. So if we want to look into what the transient states are in some of these trajectories, the transient states represent what we described as saddle nodes at the end of the previous session. So they will be equilibria which are unstable by themselves, but near which the energy is locally constant, and therefore the dynamics is slowing down. Now let's focus on this equilibrium. So you can see the slowdown right now, and then speeding up again to reach the final figure 8 configuration. So we have these saddle points from a fast initial trajectory to slow down to fast again, going to the final configuration. So possibly there could be some meaning to these equilibria. First of all, it confirms the picture that we had before, and it also might inform about some local events that the DNA may be involved in biologically. Now, recently we've started a new line of study looking at the dynamics of trefoil knots. Now, for trefoil knots, we don't have a trivial configuration that we can simply twist and perform. So we decided to start from this configuration, which is a doubly wound circle, which seems to is the one that minimizes the bending energy. And we simply impose a linking number onto that structure and then let it evolve. And we noticed so far it's an ongoing research, but we noticed some things we noticed so far is that the limiting configuration depend on the imposed linking number and ionic strength. We again find multiple local equilibria, especially for the high ionic strength. That means low electrostatic repulsion, and of course all configurations show self-contact. So we have so far an incomplete description or catalog of those configurations. Many of them, for a small amount of linking number, they look like the standard trefoil knot. For higher linking numbers, they become asymmetric. So this is for the large electrostatic repulsion. Now, when you remove when you neutralize it more, then you have other types of configurations. Plectonemes with D3 symmetry. You have the plectonemes with symmetry breaking, C2 symmetry, and you have these pretzel-type configurations, which we haven't seen before. Can be twisted, and we also found some asymmetric configurations. So we believe that this dynamical simulation in a fluid, which is very similar in a way to what other people are doing, looking at gradient flows, right? You can relate it very much what we're seeing the results to those. We think it's an important method for studying configurations of rods, charged rods in fluids, especially the DNA at high linking numbers. The benefit, perhaps, over a gradient flow would be that you can track the fluid motion and therefore obtain a deformation of the ambient space, the embedding space for the knot. And there we have some physics-based interpretation of what the energies mean and what the transitions, the transition steps and are, and some meaning, physical meaning for the slowdowns. Now the negatives of this approach is that the approach to the stable configurations is very slow, and it makes it difficult to judge whether we have actually converged to the equilibrium, and that we have configurations that are sensitive to the initial perturbation. So numerical errors as well induce other types of sensitivities. And we can only find stable configurations. So I can take questions for this part.
Yeah, David, I've got a question. Often when you're looking at DNA topology, say for site-specific recombination, you've got a single circle with two sites on it, and the problem is how do the sites come together spatially? So can you address any of these considerations in your dynamics? Is the molecule sort of cycling along its own length, or is the molecule bending in space to bring the sites together?
Right. So in these deterministic processes, you get two sites, multiple sites actually come together, as you can see in these plectonemes. But you don't have much control over what they are, and once this configuration is formed and converges to equilibrium, it becomes static. Now, in order to get, they don't move anymore. They don't slither alongside in here. In order to get that, you would need to include some thermal fluctuation in the system. So we've been working along those lines, including thermal fluctuations and seeing how the configuration may travel from one equilibrium to the other. I mean, obviously with this equilibrium, there is a whole family of those when you just slither through this configuration by moving the DNA along its length, right? Which is probably what you're thinking of. Yeah. So those motions are not happening here because we don't have any force that would propel the DNA in that particular direction. But once you add random forces, you would be able to observe those.
Okay.
David, can I ask, in this case, the delta LK that you compute, is it discounting the intrinsic rise of the trefoil knot?
This one, good question.
In case it's just an offset, but it could be there.
Yeah, this is the delta link compared to the intrinsic configuration, which is a right equal to 3. So hold on. Yeah, no, it's not. So this is actually true -5. Yeah, so the right is -3, so it's -2 as compared to the twist-free case. The twist-free case has delta link equal to -3.
Okay.
In here.
And another question, maybe I don't know if it is coming up in your last part. Are you going to cover what happens when you vary the the effective salt concentration, the screening, the electrostatic screening?
That's what I was showing here. In fact, this is the 0.1 molar. If you look on top, and the previous graph was 0.01. So this is the case where you have a low screening, and this is high screening. Okay,
so I may need to think about it, but I point out in case it's relevant. There is some work done by Mehran Kardar and co-workers some years ago. They did a very interesting study where they had a torsion-free knotted molecule with bending rigidity and excluded volume, and they showed that by tuning the screening length, you could force the knot to become either localized or delocalized, because essentially the electrostatic repulsion introduces what they call an electrostatic persistence length. And so the molecule has to decide whether it's happier to just pay electrostatic repulsive energy by localizing the tangle and keeping the rest of the chain further apart, or to spread apart as much as possible and distribute the knot evenly in the chain. So it may be relevant. I'm not quick to see it right now, but it could be relevant.
Yeah. Do you remember if they used Debye-Huckel screening or just pure electrostatic?
No, of course, for sure they had the Debye-Huckel screening. Yes, that was the point, to tune the persistence length, the screening length.
Because I can imagine if you use the pure electrostatic, not Debye-Huckel, not screen one, then I can imagine the electrostatic so big that it tries to stretch out and expand the circle and localize the knot in one particular location. Because, you know, if you just include electrostatic, the electrostatic energy is invariant under shrinkage, right? So you can pull things not to a tight knot, and it doesn't change the electrostatic if you don't have the screening component. So I can imagine that might be the case. Okay. So I'm going to go to the last part now, where I'm focusing more on the biological role of DNA and how supercoiling is reflected in that. So the main processes, of course, that DNA undergoes in cell are the replication, transcription, recombination, and in eukaryotic cells, chromatin remodeling. And the replication is simply taking DNA and making another copy of it. During cell duplication, transcription has to do with the expression of various genes, and the DNA bends there because it's bound to proteins that regulate transcription. Now, in recombination, the DNA bends because various parts of the DNA with replication sites need to be brought together in order for the DNA to be reconnected in those sites, and that leads to DNA bending. And in chromatin, the DNA is wrapped around these histone core particles on various places, and that wrapping and forms various loops within the chromatin core that have energetic consequences. So the DNA, of course, has the biologists think of DNA as a DNA code that codes for proteins, but from our perspective, we want to think about a different code that we can call regulatory code, which directs the regulation of gene expression. And there is a regulator. There are some pieces in DNA where proteins bind, DNA binding sites, but there is also pieces of DNA between those sites which deform and form a loop, and the DNA in those pieces, the deformation of that DNA will have a strong effect on what the resulting function during transcription is. So just to make it simpler, we're going to look at just a simple problem with one protein with two binding sites and a DNA in between which deforms into a loop. And it has been found that DNA does not, unfortunately, behave like the idealized structures we've been talking so far. The elasticity is locally variable, and there are different types and modes of deformation that one has to account for, not just pure bending and twisting. These things can be coupled. So in order to account for that, one can develop a different model, which is geometrically different from the rod type models that we've seen so far. And in here, the DNA base pairs are in fact represented as rigid building blocks, rigid base pairs, rectangular blocks. And now we want to express their kinematics using these angular variables, tilt, roll, twist, and also the position variables shift, slide, and rise, which describe the relative position and orientation of two neighboring base pairs. And then we can express the energy of the structure by summing the energy of individual base pair steps. And those local energies then will be expressed as functions of the six parameters here. Now it has been, if we just look at the quadratic approximation of this energy, we already have a rather complex picture of what DNA does. There are segments which prefer to be straight, like for example base pairs AA. A sequence GG prefers to be bent. So if you combine five base pairs of A and a few base pairs of G's and C's, you get so-called A tracts, which overall exhibit intrinsic curvature. Now there are also base pairs which exhibit twist-roll coupling, so there would be coupling between the twisting and bending deformation. Twisting and stretching is coupled as well, so if you stretch the DNA, it has a tendency to over-twist. There's bending and isotropy, so the DNA is more flexible in one direction than in the other to bending, and there's some shearing deformation as well. So one way we can get a handle on the constants describing this is by analyzing crystal structure, and Wilma Olson has done that type of analysis, or there are researchers who do molecular dynamic simulations of short DNA segments, Richard Lavery and John Maddox, and they obtain also estimates of these bending and moduli, twisting moduli, and natural position constants. So from those, we can then construct a model for DNA deformations that will reflect real structures found inside cells. And in order to make them realistic, we need to include also structures of proteins. So, for example, cap protein, which bends the DNA when it binds, or LAC protein, which I'm going to talk about today. LAC is a molecule which binds DNA in two different places, so it has two DNA binding domains, and then it can form a loop. And the LAC plays a big role in the repression of a very famous operon called the LAC operon in E. coli, which has been studied extensively. The LAC repressor is a protein that binds to two different sites within the operon. There are three such sites, in fact. There's O1, O2, and O3. We're going to be looking at the O1 and O3 sites, which are near each other, and how the LAC binds to it. It has been found experimentally that if you change the distance of these two sites, you affect the repression, the effectiveness of repression of transcription by the LAC up to 30-fold. And how does that repression, how is that affected? It's affected in this periodic fashion. So the number here would be distance between the two sites, and as you change the distance, the repression rises. So that means no genes are expressed in the peak. A lot of the gene is expressed at the prow, and again, no gene is expressed at 70 for the distance 70. A lot is expressed around 75. So you get a roughly 10.5 base pair periodicity here of repression efficiency as a function of the operator distance. And you can do various types of experiments to confirm that. These are older experiments on constructs. There are newer experiments as well. So the question is, how does one explain this periodicity and also the efficiency of the repression, which is small for short loops and then goes up and then decays and then relaxes again. So there's a size of the loop around 70 base pairs, which is the most efficient for repression. So the way we want to do that is we want to associate the efficiency of repression to the energy of the loop that is formed between the two sites. So in order to compute the energy of that loop, we need a sequence for the loop, which will determine its energetics, and we need the structure of the protein, and then we also need to analyze the topology of attachment of the loop. We call it looping mode to the protein. There are 4 different types of loops, and there is also a flexible protein state that one can look that is possible for this protein. So taking all this into account, we've been able to determine the energies of the different types of loops, either for the V-shaped protein or for the flexible protein, and we found that the flexible protein has the smallest energy for the bound DNA. And then when we looked at how that energy depends on the spacing between the sites, we found that the periodicity of the spacing, we looked at all of these different types of loops, and we found that the open configuration of the protein gives you the best agreement between the spacing of the loop and the locations of the least and the largest expression. And from that, one can conclude that the open configuration of the repressor is the one that is most likely to be found, and that's the one that controls the expression of the gene. So this is just an example of how we can use the mechanics and the ability of compute the mechanics of the DNA structure in addressing an actual biological question and results of an experiment. So we used DNA elasticity and electrostatics to analyze these configurations, and we found that the loops are formed with the extended conformation. Now the challenges here are that even though we try to include as much realism as we could into the calculation, there are still unexplored venues. There are proteins that bind to DNA other than just the repressor in vitro. And also there are difficulties with computing these configurations and computing the free energies of those states that have to do with computational expense. So to conclude here, I would like to just thank all the people that I have collaborated with on these various projects, from my PhD advisor, Bernard Coleman, my postdoc advisor, Wilma Olsen, another of my advisors, Erwin Tobias, and then collaborators, including students, Luke Tapla, Grossner, Yoav Bitton, and then Suk Lim, my collaborator from University of Cincinnati, with whom I worked on the dynamical model, Attila with Kochish, with whom I worked on the DNA mechanical experiments, and Abby Pekoski, my current student, PhD student, who's working on the dynamics of trefoil knots. And I also want to thank to the funding agencies for supporting our research. So I can
Very
take
good,
questions now.
David.
Mhm.
Thank you very much. Very interesting. I have a basic question, so then I leave to others more technical questions. Is this model made of plaquette? I don't know if you have a name for that. Going to really be the future or the next step is certainly a next step, but leaving behind, so to speak, the typical rod model that evidently shows limitations. You can take account of all these effects, roll and tilt, etc., etc. So, is this going to be the model, the basic reference model for the future years to come, or what do you
I'm
think?
hesitant to say that because I believe the continuum models have their benefits. Simply by treating the axis continuously, you could do analysis with them. So I would say these two models can easily live in parallel. Each has its pluses and minuses, and I think we should explore both of them. One can use the discrete model to define linking number and look at some geometrical questions. I'm not sure that it's always beneficial in all the situations. I think would be my answer. I think both of the models would be good to be worked on.
Okay. Thank you.